Hello, everybody. Welcome to a new episode of the Dissenter. I'm your host, Ricardo Lopes, as always, and today we're joined again by Dr. Henry Greely. He's the Deane F and Kate Edelman Johnson Professor of Law and Professor by courtesy of Genetics at Stanford University. Last time we talked about the end of sex, and today we're going to talk about his latest book, CRISPR People, The Science and Ethics of Editing Humans. So, Dr. Greely, welcome to the show. It's a pleasure to everyone.
Thanks. It's nice to be back with you.
Okay, great. So, first of all, could you please explain briefly what is the CRISPR technology and how it works?
Sure. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is why it's really good that there's an acronym for it that is easily pronounceable. It's become CRISPR. It's also a particularly nice acronym because, at least in English, you can easily turn it into a verb or an adjective or an adverb. You can CRISPR people. You can do things CRISPRly. It's a good it's a good acronym, and I don't often think of acronyms as good things. It is a bacterial defense system invented by bacteria 3 billion years ago or so. It is a way for bacteria to try to attack invading viruses. It turns out that viruses are not just plagues for humans. They don't just cause pandemics in humans. The viruses will attack just about anything, including bacteria. Bacteria do not have the kind of immune systems we have. I don't know that you could vaccinate bacteria, but much to scientists' surprise, it turns out that they do have a variety of defenses, one of which was uncovered in the space of 20 years or more by various scientists just doing natural history research. Just trying to figure out what this odd DNA and RNA and proteins, what these strange things were in some bacteria. Some of the scientists actually worked for a yogurt company because some of the yeasts they needed and some of the other microbes they used in yogurt production were being affected by this. So scientists, and particularly a Spanish scientist named Francisco Mojica, were instrumental in figuring out that CRISPR was a way bacteria used to cut up invading viruses. It became much more interesting in 2012 when several groups, the first led by Emmanuel Charpentier, who was then at Umeå University in Sweden, and Jennifer Doudna at University of California at Berkeley, published in Science a way to use CRISPR as a tool. And it's CRISPR as a tool, not CRISPR as a natural bacterial immune system, that has gotten all the attention. I think the best way to think about it is as a word processor function. If you use a word processor, you can have it find something. So let's say somebody has written something that has my name in it. My name is spelled Greely, but most people with my name spell it Greeley. I can set my word processor to find every place in the document where it's spelled Greeley. Part of CRISPR is that locator. It uses RNA to find pieces of DNA that have specific sequences, that have specific letters, just like Greeley. The CRISPR then uses an associated protein to cut that found piece of text out. To cut that DNA out, in the word processor analogy, it would be like using a cut function. So with, say, Microsoft Word, I find everywhere that it's Greeley, and I cut it out. You can add on to CRISPR a replace function, where the CRISPR construct not only has the RNA that locates the sequence you're interested in, and the protein that cuts out that sequence, but also has associated with it a chunk of DNA that you hope that often, sometimes usually, will take the place of the cutout area. And so that would be like telling Microsoft Word, find everywhere in this document where it's Greeley, cut it out and replace it with Greely. Now, I'm not a huge fan of Microsoft Word. It's more efficient than CRISPR, at least when it comes to the replace function. But CRISPR is more efficient than anything else we've ever done before. And what's made it revolutionary is not that it allows us to edit genes. We've been able to edit genes certainly for the last 10 or 15 years through a couple of systems called TALENs and zinc finger nucleases, but arguably since 1971 with recombinant DNA, CRISPR makes it faster, easier, cheaper, more effective than the alternatives. The example I like to use, the analogy I like to use is in automobiles. In the United States, there were automobiles before 1910. They were rare. They were expensive. They broke down all the time. You needed to be rich to buy one and to hire the chauffeur mechanic who would keep it running. Then Henry Ford came out with the Model T. And suddenly cars were cheap and reliable. And you didn't need a chauffeur mechanic on staff. And suddenly, cars went from being rare to being pretty common and then to being ubiquitous. So CRISPR is revolutionary not so much because of what it does, but because of how easily it does it, how cheaply, how quickly, how effectively compared to previous alternatives. In 20 years, we probably won't be using CRISPR-Cas9, just as people don't drive Model Ts today, but we will be using its descendants or relatives of it to do gene editing even more effectively, efficiently, easily, and cheaply than we can now.
Mm-hmm. But is CRISPR already widely available to people in general?
CRISPR is being used in high schools in the United States, in high school biology classes. It is widely available. It's being used in tens of thousands of laboratories around the world. From the moment it was introduced, it was one of the most rapidly picked up scientific tools anyone can remember. I remember talking to a scientist friend of mine at Stanford, probably in 2013. And I said, so, hey, has your lab tried this CRISPR stuff? And he said, yeah. Well, how's it gone? Well, it's a new tool. I expected it would take us a month or two to figure out its pluses and minuses. And it took us less than one afternoon. So its simplicity has made it widely adopted. It's used in laboratories all over the world. It turns out to be a multi-use tool. This use for identifying, locking onto, cutting out, maybe replacing DNA sequence is the most famous and right now at least the most important use of CRISPR. But particularly its ability to find and lock onto bits of DNA mean that it can be used for other things. So for example, people are working on using CRISPR for very rapid, easy at-home tests for COVID infection. Mm-hmm. There it's not looking for DNA, it's looking for RNA, but it can look for either one. And you put some COVID construct on a strip, put some fluid, some saliva from a patient on it, and the idea is any of the RNA from the virus will stick to the COVID and cause, and then through other operations, cause the strip to change colors. I don't think anyone has gotten approval for that yet, but lots of people are working on it. It turns out that COVID, to use yet another analogy, will be kind of the Swiss Army knife of tools. Its main use is the cutting, but it also includes a corkscrew, a tweezers, a magnifying glass, and all the other things you get in the fanciest of the multi-purpose pocket knives.
Right. And has it already been used in humans?
So CRISPR in humans, certainly CRISPR is being used enormously in research with human cells outside the body. Uh-huh. So it's quite easy to use CRISPR to take a human cell line and make specific modifications in it to change one gene to a different variation to see how that affects the function of the cell or the success of the cell line. In humans, it's being used on a experimental basis as a treatment for genetic diseases. And sometimes it's being explored as treatments for non-genetic diseases as well, as ways to say, well, non-genetic, non-inherited genetic diseases. Cancer, for example, is always a genetic disease in that it's a disease of the genes. It's caused by mutations, but those mutations are only rarely inherited mutations. They're acquired mutations. So CRISPR is being used to try to treat genetic diseases, other diseases with genetic components, and some diseases that have no genetic components at all. I do not believe any CRISPR therapeutics have yet been approved anywhere, but they're getting close. And there's certainly CRISPR therapeutics that are in the phase 2 and phase 3 clinical trial process. I expect that there will be at least some gene therapies approved using CRISPR before long. Now, there's an important distinction here in using it in living people as a gene therapy, as a treatment. One way to do it, the easier way to do it, which you can do with certain diseases, is by taking human cells out of the body, CRISPRing them, editing them outside the body, checking to make sure you've made the appropriate edits, that enough of the cells have taken up the edit, that you don't have unwanted edits, and then putting those cells back. For blood diseases, that is a very plausible alternative, taking the blood-forming stem cells out of the bone marrow primarily, modifying them in the lab, making sure that you've made the change you want, and then putting them back in. So things like beta thalassemia or sickle cell anemia or a wide variety of other blood-based diseases are potentially susceptible to gene therapy with the CRISPRing being done ex vivo, outside the body. Tougher, but people are working on this as well, is using CRISPR inside the body, having some kind of vector, some kind of delivery vehicle that will take the CRISPR to the cells that need it, insert it there, have enough uptake, and treat the disease. An example of a disease that might be treated that way is cystic fibrosis, where the main effects, it's a genetic disease. All the cells of a patient's body have the same problem, but most of the cells, it's not very important. It's mainly important in lung cells and also important in pancreatic cells. In men, it affects sperm production. So there are a few organs where it's relevant. One could imagine, let's say with lung cells, putting CRISPR into an inhaler, like an asthma inhaler. You take a deep breath, the CRISPR construct goes into the lungs, finds the lung cells, inserts itself into the lung cells, makes the changes in the DNA in the lung cells, and somebody's healthy. That's a great goal. People are working toward it. It's trickier to do it in the body than outside the body because it's harder to do, for example, the quality control, to make sure you've made the right change and not the wrong change, and to make sure you've done it in enough of the cells. But that's how CRISPR is currently being explored, and I think it's going to turn out to be explored very, very successfully for treating diseases. The most important thing for many purposes about that is if I treat your genes in, say, your lungs or your bone marrow, and I change your DNA, and you're healthy, we're all happy, but that change dies with you. Your children do not inherit that change unless I have changed your sperm. Now, normally I'm not going to change your sperm, at least not on purpose. One thing people do have to worry about a little bit is that the gene therapy aimed at the body cells, the so-called somatic gene therapy aimed at the soma, the body, might also affect some of the reproductive cells, the eggs or the sperm. But that's unlikely and can be protected against. The other kind of CRISPR editing on humans, and the one that has been much more controversial, is CRISPR editing that includes the germ cells, the gametes, the eggs, and the sperm, because that not only affects you, but could affect any children you have, grandchildren, great-grandchildren, could in theory ultimately affect the entire human species. Or not. You might not have children. Your children might not have children. Their children's children might not inherit the edited gene. So it's not what sometimes people get carried away and assume that this will necessarily affect all of humanity in the future. It may not, but it has the potential to do that in a way that somatic cell gene therapy does not. And that has many people concerned.
Mm-hmm. What about that experiment from 2018? You focus a lot on the book, the He Jiankui. I hope this is the correct pronunciation of the name.
It's not. I have tried with Chinese speakers to perfect my own pronunciation of it, and it's close to even repeating from a patient person. I can't get it right. It's something like He Jiankui, and that's wrong too. But let's just call him He and leave it at that.
Yeah.
Most of the book is the book was certainly inspired by the He Jiankui experiment, or maybe a better word for it would be fiasco, because I do not think it was a good experiment at all. About the first half of the book is really sort of a narrative of what we know about He, about the experiment, about the discussions about ethics before the experiment, about the world reaction to the experiment, about who knew what about the experiment when. It's a story, and I think it has a good narrative flow and is, I think, nicely readable. One problem with it is we're not sure any of it's true. We have very, very little independent confirmation of anything that happened. Mainly, we've got what He says, what his collaborators say, and what the Chinese government, through a handful of press releases, has said that it's found. And we've got no independent outside proof that the babies even exist, let alone whether they're healthy and what changes were made. But taking at face value the information we have, in November of 2018, He, pushed by the premature, from his perspective, release of a story by a reporter named Antonio Regalado in the MIT Tech Review, announced to the world at a summit in Hong Kong on human gene editing that 2 babies had been born whose DNA he had edited in embryo, that he had edited that DNA to try to knock out, to make ineffective, a gene called CCR5 that normally produces a protein, happily also called CCR5. CCR5 protein turns out to be important for the most common strains of HIV to infect T cells. You can still get become HIV infected even if you have no CCR5 protein, but you're substantially protected. Infection becomes a much, much lower possibility. We know that because there are humans among us who have no functioning CCR5 gene. This variation is most common in Northern Europe. About 1% of Northern Europeans don't make any CCR5. We actually don't know what the effects are on their health long term. We do know that there are people with that variation who live healthily into their 50s, 60s, and 70s. We don't know whether more of them die off earlier, whether it's protective. There's a little bit of preliminary evidence that the absence of that protein makes you more susceptible to bad illness from influenza or from West Nile virus, but it's very preliminary work. The other thing we can say for sure is some people with this unusual variation do manage to survive in a healthy state late into life, and they are largely resistant, largely immune to infection with HIV. Mm-hmm. So He tried to make these babies resistant to HIV. He announced in November of 2018 that he had successfully done so. It turned out in the following year that there was a 3rd baby born in the summer of 2019 which had also been edited by He as an embryo. We know nothing about that baby. We just know there's one sentence in a Chinese government press release that tells us that such a baby exists. So that was the catalyst for lots of concern, the catalyst for lots of public attention, and ultimately the catalyst for the book. Because He was modifying the DNA in the embryos, he modified not only the DNA that would lead to the T cells, which were the subject he was most interested in, preventing the T cells from getting infected with HIV, but also brain cells, liver cells, heart cells, and in the case of these two little girls, egg cells. So this is both somatic cell gene editing and germline gene editing. And in theory, if these babies were in fact edited, they could pass that along to their descendants.
Mm-hmm. So in this case, if it happened, would it constitute a form of human enhancement?
That's a really good question. We like to think of treatment and enhancement as these two different things. But like many things, the separation isn't as nice and crisp and clean as we like to think. And preventing disease falls somewhere in between. It's sort of like the COVID-19 vaccine I got in February. It prevents a disease, but it doesn't treat a disease. It enhances my immune system, but it's related to my health, not to my ability to play sports or do math or do crossword puzzles or the other things, or my eye color, the other things we think of as enhancement. So there are some things that are sort of in between treatment on the one hand and enhancement on the other. They're actually sort of medical enhancements might be the best way to think about them, which gives them some of the positive virtues of a medical intervention, as well as some of the negative aspects of enhancing. This I would call a medical enhancement, something akin to a vaccine. It's a genetic preventative intervention.
Mm-hmm. So if, for example, China was successful in terms of editing the genome of fetuses, for example, or perhaps in the case of germline cells, do you think that in the future there could be some sort of arms race between countries to develop this technology further?
I think that different countries will be very interested in developing these technologies for a variety of reasons, but I don't think producing enhanced citizens is likely to be part of it. I sort of like the idea of an arms race, I can imagine. So China makes people with 4 arms, and Germany makes people with 6 arms, and the US makes people with 8 arms, an arms race quite literally. The problem with the idea of using, it is this enhancement idea, making people different in ways that make them, for at least some purposes, better. That is, I think, behind most of the public concern, and not just public, most of the professional concern as well, about gene editing. There's a fear of dystopian science fiction novels coming true, where there's a genetically enhanced upper class or upper caste, and a genetically inferior lumpen DNA proletariat. There are even fears that this could lead to a speciation event where the upper class can't interbreed with the lower class. I think those fears are exaggerated, and at least for the next several decades, highly implausible. We don't know how to enhance people genetically. We just don't know how to do it. If you look at something as relatively straightforward as height, we know of hundreds of genes where the particular variations in the genes play some role in how tall a person will be. If you add together the expected effects of all of those genes, you explain about 15 to 20% of the variation. Some people would say as much as 30% of the variation in adult height. If you look at the adult height of a person's parents, and then you ask whether the person is male or female, those data points can explain about 90% of the variation in height. So even with something as straightforward as height, if we wanted to make people taller, maybe we could pick 15, 20, 30 genes and edit them to a taller version, and maybe we'd get a centimeter of additional height. Maybe we wouldn't. Maybe doing that would cause other risks and harms to people. Maybe it wouldn't. The unknowns here are enormous, and that's with something relatively straightforward. If you get into what people are mainly worried about, I think, things like intelligence and other behavioral characteristics, making the perfect athlete, making the perfect warrior, making the perfect mathematician, making the perfect podcast interviewer, obviously something countries will certainly want to do, make that perfect podcast interviewer. We just don't have a clue what the genes, what the genetic, we have only the faintest clue of what genetics will be involved, and we are confident that it's a lot. So let's take intelligence, leaving aside for the moment the fact that we can't even define it. There's no agreed upon definition of what intelligence is, or even whether there's one sort of generalized intelligence versus a variety of different cognitive skills. We know of literally hundreds of genes where variations can have an enormous effect on intelligence, and that if the gene is broken, your intelligence is very low. We know of no genes where the variants make a significantly can have an effect of substantially increasing your intelligence. The people who are most optimistic about progress in this area, more optimistic than I am, but enthusiastic that they think they've found things, think that by looking at 100 different genetic variations, they can predict 1 or 2 IQ points. Now, IQ's baseline is 100. How worried am I about a country having an average IQ of 102 versus an average IQ of 100? Not very, particularly since we know that there are a whole bunch of non-genetic things that can increase IQ test results, like good childhood health and nutrition and good parenting. There is this interesting phenomenon called the Flynn effect, where over decades, populations' IQs go up. Every 20 years or so, the IQ test gets rescaled because it's supposed to center at 100, but after about 20 years, the average person is scoring 110. So they rescale it back down to 100. We don't know why populations are doing better on IQ tests, but it's not because their genes are changing. So I'm not very worried about the enhancement issues, certainly not for my lifetime, probably not for your lifetime. If I have grandchildren, maybe in their lifetime, maybe not. And brains make genes look simple. Brains are so complicated, 89 billion neurons, each of which has on average about 100 connections. Each one of those connections, called a synapse, is not like an off and on transistor. It's like its own independent analog computer, getting a variety of different neurotransmitters where the timing and the quantity and the nature of the neurotransmitter all make a difference. They're affected by genes, yes. There is a genetic contribution to cognitive processes, but they're also affected by environment, and they're also substantially affected by chance. In human development, neurons are genetically programmed to move in particular directions, to grow their wiring, their axons toward particular regions. But where exactly they land is more a matter of chance than anything else. There's really interesting work being done on this by an Irish geneticist whose name escapes me right now, but he has an excellent book about the complexities and the roles of environment and chance in brain development. They're huge. Will we ever fully understand it? Not clear to me that we will ever fully understand it. Will we ever understand it as being fully explicable by genetic traits? That I'm pretty confident we will not. It will not turn out to be fully explicable by genes. To the extent genetic variations play a role, it's going to be a very complicated role involving thousands of different genetic variations. Not in my lifetime or in yours.
Yeah, but even if these applications, when it comes to some more complex traits in humans, are not really around the corner, do you think that we should already be worrying about the ethics of gene editing?
I think we should, yes. I think we should be considering the ethics of gene editing. Not sure I agree with the verb worrying. We should be paying attention to it. We shouldn't be panicked about it. But, ethics encompasses a lot of things. We've been talking thus far about the long-term social and ethical effects of really designing babies, of making babies with particular traits through gene editing. That's why this book, which is about gene editing, is different from my first book, The End of Sex, which is about embryo selection. Two parents mix their genes in an embryo, in 10 embryos and 100 embryos, and they pick the best of those, the one they like best out of that. This is not selecting from what the parents have. This is adding things, and that gives it the ability to do multi-gene, to pick particular variations in many genes in a way that gene selection, embryo selection would not. We've been worried about these broader ethics, but there are some other really important ethics, and that's what I mainly condemn the He fiasco for. It's not the idea that there will be enhanced super races in the future, because I think that's highly unlikely. It's because, first, this was a recklessly dangerous intervention on these embryos for no good reason. A fundamental issue in human subjects research is that the risks have to be justified by the potential benefits either to the subject or to science. Nazi doctors were hanged by the neck until dead in the aftermath of World War II for unethical experiments, and one of the things that was unethical about them was a terrible risk-benefit ratio. The fact that they involved murder and torture were also unethical aspects of those experiments. Whose experiment is fundamentally deeply unethical because he exposed these embryos to huge risks. He'd never done this successfully in anything else, not in monkeys, not even what he was trying to do in mice, with the potential benefit to them. Well, maybe they would end up being more resistant to HIV, which they probably would never get exposed to for at least 20 years, and maybe not then, at which point there are already other ways to avoid infection, and in 20 years there will probably be still other ways to avoid infection. Benefits to them were minor. Benefits to science were minor at best, and in fact one of them, according to He himself, won't even be partially immune from HIV infection because he only managed to edit one of the two chromosomes on which the CCR5 gene is carried. The potential benefits to the babies were tiny, and for one of the two babies, really non-existent, because at least according to his own data, He was only able to change one of the two chromosomes on which the CCR5 gene exists in one of the babies. So in one of the babies, both chromosomes were changed, and the CCR5 gene was knocked out. In the other baby, only one of the genes on one of the two chromosomes was changed, which means that baby will still produce CCR5, will still have CCR5 on her T cells, and will still be susceptible to HIV infection. No benefit to her, and he knew about that before he moved the embryo into a uterus for possible birth. There are also risks that we don't know about in terms of what is it like to live without CCR5. The people in mainly, but not exclusively, Northern Europe who naturally have it knocked out all have the same variation. A set of 32 letters is knocked out of their gene. That's what He tried to do in these embryos, but he didn't succeed. He made different mutations in the embryos, mutations we've never seen before in humans. And we also don't know whether lacking CCR5 will put these babies at some higher risk for other things, maybe things that are more common in China than they are in, say, Finland. We don't know. It is at least worrisome that CCR5 seems to be found in all mammals, birds, reptiles, and amphibians. So the fact that it is so widely conserved seems to indicate it's doing something useful. We just don't know what.
So let's talk now a little bit about the legal aspects of gene editing. Is there anything in the law, and in this case perhaps we can focus on the US or other countries if you want, that prohibits gene editing?
Yes, no, and maybe. It's important here, I think, to distinguish between the somatic gene editing, gene therapy in living people that doesn't go down to the next generation, and the germline therapy. In basically every developed country, the somatic gene therapy is regulated the same way, well, in ways similar to the ways that drugs and medical devices and biological products are regulated. So in the US, you'd have to get the approval of the FDA. In Europe, the European Medicines Agency would have to approve, and similarly. And they're looking at the safety and efficacy of the treatment. So in the somatic cell sense, it is regulated in a relatively routine way, as I think is appropriate, like many other medical interventions. Germline is a more complicated story. There are some countries that explicitly ban it. The United Kingdom has made it illegal in 1990. Some, but not all, European countries have made it illegal. The Council of Europe's Oviedo Convention on Bioethics says that it is illegal, but that requires individual countries to both sign on to that convention and then pass implementing legislation. Some countries have, some countries haven't. In China, there was nothing that clearly made it illegal, but after the He story broke, Beijing decided it was illegal. And I'm not an expert in Chinese law, but my strong impression is whatever the statute says, when the government decides something is illegal, it's illegal. And He, in fact, was sentenced to 3 years in prison as a result of criminal charges brought against him. And the United States is a particularly interesting and strange example, as it is in so many ways. There's no law in the US that says germline genome editing in humans is illegal. But our Food and Drug Administration, our FDA, has taken the position that any alteration in human genes falls within its jurisdiction as either involving a drug or a biological product. And so in order to do it, you have to get FDA permission. In order to do it on a clinical basis, you have to prove to the FDA that it's safe and effective. But the FDA will let people do research in humans. FDA doesn't care about research in human cells or in vitro in plastic, but it does care about research done in humans, clinical trials. You can do clinical trials with something called an investigative new drug exemption, where you just have to show, well, we've got some reason to think it's kind of safe and that it might work. If you wanted to try this in the US, you'd have to get one of these INDs from our FDA. The FDA is likely to be, for both scientific and political reasons, very cautious about giving such an IND for such a controversial intervention. But our Congress took it out of their hands, and in 2015, starting in 2015, has every year put into the appropriations bill, the bill that allocates money to the FDA, a bill that has to be repassed every year, a provision saying none of this money can be used for to deal with research involving heritable human genome changes. And furthermore, that no application to do that work can be deemed received by FDA. And there's a funny little quirky law story behind that. Under FDA law, if you as a researcher apply for one of these INDs to let you do research in humans, and FDA says nothing for 30 business days, then it's approved. FDA needs to block it. Well, the problem, the key issue there is it's 30 days after the FDA receives it. The statute says even if you got the entire Supreme Court to testify that they saw you hand it to the receptionist at the FDA, it doesn't count as received. And if it never is received, the 30 days never starts. So right now, it could not legally be done because it requires an IND, and FDA is not able to give an IND even in the unlikely event that it wanted to. But that's a year-by-year piece of legislation, and if it's not readopted for the next year, it goes away. So yes, there are a lot of countries that make this illegal in a variety of ways, but there are also an awful lot of countries that just don't deal with it at all. And kind of for understandable reasons. If you're Paraguay or the Central African Republic or Laos, you probably have more important issues to deal with than human germline gene editing.
Mm-hmm. Are there any other technologies that are nowadays already socially and morally acceptable and are legal, for which we already have a sort of history of how the legal system dealt with them, to which we can compare CRISPR and gene editing more generally?
Yeah, I think so. I would say one of the nice examples is in vitro fertilization. Mm-hmm. First human baby was born through IVF in 1978. Louise Brown, who's now 42. She will, we trust, turn 43 in July. That was extremely controversial when it first happened. Different countries scrambled to figure out what the laws should be, how it should be regulated. And countries have come up still with very different laws, from very restrictive to very welcoming. One of the intriguing things about this, it's a nice example of the complexity of the law. Sometimes the laws just say you may or may not do this. Sometimes in countries, I would say civilized developed countries, unlike my country, where healthcare is provided to everyone, it's done not so much through you may or may not do this, but our healthcare system will pay for this but not that. So in some countries, healthcare systems will not pay for IVF if it's done by an unmarried couple or by a single woman, but it will pay for it if it's done by a heterosexual couple or a married couple, etc. Regulation through the healthcare financing system. So different countries have come to different answers. And there's still a whole bunch of questions that remain kind of up in the air. The whole role of surrogacy, which would not exist in its current form, but for IVF, continues to be controversial and changing and not entirely clear in some countries. So I think IVF provides a nice example of how it has worked. It is not necessarily a nice example of how we would like it to work. I think in retrospect, we would have been better off paying attention to IVF in advance. IVF was a great shock to people. People didn't expect it. There hadn't been statutes proposed dealing with it. The UK responded in 1990 with the Human Fertilization and Embryology Act, which did govern it in, I think, a very interesting way and in a way that looked forward to other possible future actions like human gene editing. And I think the UK, to my taste, may have done the best job at this. The US has basically left it almost entirely unregulated, which has some advantages, but also some disadvantages. So I think that's an example, and it's a nice example because it does touch on these issues of reproduction, families, that people care so much about. An earlier version is just artificial insemination. When artificial insemination first happened, there was uncertainty about whether the babies would be viewed as legitimate or not, whether or not they could inherit, all sorts of different issues that nobody had thought about. So I believe that it is useful for us to think about some of the gene editing issues, but it's also useful for us not to exaggerate the dramatic change, to take too exaggerated view of just how dramatically it's likely to change the world, because I think it's not likely to be that important. It's actually so the first half of my book is about the story of and the lead-in and the ethics. The second half is an assessment of both the He experiment and then more broadly the idea of human germline genome editing, where I reach a somewhat unsatisfactory conclusion in the sense that I'm kind of in the middle, so I'll get shot at from both sides. I don't think there's anything inherently wrong or evil about germline modification. There is no such thing as the human germline genome, like the Ark of the Covenant from the Indiana Jones movies. There are 7.5 billion different human germline genomes, and they change each generation, and human action influences the directions in which they change. Our genomes are different from those of our hunter-gatherer ancestors because of agriculture. Our genomes are different from our great-great-grandparents because medicine has allowed some people who would otherwise be dead because of genetic reasons to survive and reproduce. So I don't think there's anything inherently wrong with it, but on the other hand, I don't think there are very many good uses for it. Through pre-implantation genetic diagnosis, the technology I mainly talk about in The End of Sex, if you're interested in avoiding a particular single gene problem, you can do it by picking embryos, almost always. You don't need to edit embryos. If you are interested in that, you may also be able to fix it after the baby is born through the increasingly good uses of gene therapy. I think there are a few situations where if a couple wants to have healthy kids that are genetically, quote, theirs, close quote, editing is the only way to do it. If a couple has the same autosomal recessive disease, so cystic fibrosis, sickle cell anemia, beta thalassemia, each person has 2 copies of the form of the gene that causes the disease. Let's say 2 people with cystic fibrosis live long enough, are healthy enough, fall in love, want to have kids, but they'd rather have kids without cystic fibrosis. Any child they conceived from their own eggs and sperm would have to have cystic fibrosis, because the only genes they can give that child are genes that are predisposed to cystic fibrosis. They each have 2 copies, and both of the copies are disease copies. For them, the only way to have a child that is, mainly, almost entirely genetically theirs, fibrosis, and then fix it in therapy after birth, not possible yet, but maybe soon, or edit the embryo. I don't see any really good, compelling reason to say those people shouldn't be able to have healthy kids. On the other hand, how many people like that are there in the world? More than 0. Probably more than 100 or more than 1,000, but probably not more than 100,000. That's probably a high estimate of the number of couples who would face that. So I just don't think it's going to be very useful, very important, unless or until we know enough to do enhancement, which I think is not in the cards anytime soon. But on the other hand, I don't think it's deeply important, deeply wrong. So people who think it's deeply wrong and shouldn't ever be done are mad at me. The people who think this is going to be a really important medical breakthrough are mad at me. The advantage that way is I know I have to watch for arrows coming from every direction because I'm stuck in the middle.
So just one last question. do you think scientists should be careful about how they communicate information about CRISPR and gene editing to the general public?
Yes. So you broke up a little bit asking the question. You may need to re-record the question, but I think it's crucial for scientists to pay a lot of attention to how information is conveyed to the public. I think more fundamentally, it's also important, crucial for scientists to pay attention to the public. So one problem is often there's a scientific paper that has potentially exciting results. The journalists will often, not always, good science journalists are wonderful, but there are a lot of not so good journalists on science. And it's exacerbated by the headline writers who are never the journalists. They will play it up in order to get more readers and make it more scary and more threatening than it otherwise is. And in a sense, it's also a problem within the scientific institutions themselves. Most research institutions now have communications or press offices, and part of their job is every week put out a list of all the great new breakthroughs that Stanford scientists have come up with in the last week. It's marketing. But that marketing, these great breakthroughs, also give people the impression that things are much farther along and much more dramatic than they actually are. If 1 in 100 of the breakthroughs that gets announced every year actually turned out to affect the world, that would be a surprisingly high result. So, yeah, I think people need to be very careful about how things are presented to the public to make sure that they're presented both with their limitations, saying, yes, this could potentially, at some point in the distant future, it might lead to this exciting result, but it also might not. But more deeply, and I see this and I criticize it a little in the germline discussion, scientists, science with a capital S, sometimes gives the impression to the rest of the world that it should be the only ones that regulate it. Science should only regulate itself, and the outside world should let science do what science wants because science knows best. I quote one person who, loosely, and I think he regrets it, about how, well, science will let the rest of the world know when this should be used, when this can be used. Science cannot be divorced from the societies it lives in. It shouldn't be as a matter of political theory. And no country has adopted the Platonic idea of philosopher kings, but substituting scientists for the philosophers, or even bioethicists for that matter. I'm a Democrat with a little d. I think democratic representative government is the best, the least bad form of government around, the best form of government around. And in that theory, scientists should inform the decisions, but scientists don't make the decisions. They provide information that may be crucial to making the decisions, but ultimately the decisions are made by the people acting through their appropriate government. Science needs to be humble about that and needs to regularly say, you know, the consensus within our community is this is fine, but we recognize that if the people don't want it, then we won't do it. And humility is one of those virtues that's often in short supply. I think science needs to not only communicate well, science needs to also communicate in advance to avoid surprising people. So if you tell people well enough in advance that what's going on and why you're doing it and what the good reasons are that you're doing it, and you're not Victor Frankenstein doing it because it's exciting and fun or you're obsessed by it. But then in the longest run, science needs to accept that it doesn't make the rules, society makes the rules, which in turn should make it even more, make it clear, make the importance of proper communication to society even clearer.
Mm-hmm. Let's end on that note then. And again, the book is CRISPR People, The Science and Ethics of Editing Humans. Oh, there it is.
So not yet available in a Portuguese translation, although I live in hope.
Yeah, I always read the books in English, so I mean, I myself don't have that problem, but yeah, for the Portuguese public, it would be nice to have a Portuguese translation. So, just before we go, would you like to mention any places on the internet where people can find your work?
Um, I have a website at my law school that lists some of my publications. There's a link there that lists all of my publications. I'm not sure I recommend that, but it lists some of they also sort of pull out some of the more recent ones. And it has a bio that talks a little bit about the various areas I work in. I am dilettante is maybe too strong a word, but I don't focus just on one set of bioethics issues. I work on neuroscience, genetics, assisted reproduction, stem cell research, and then other weird, interesting stuff that comes my way, like de-extinction. So if there's a bioethics related topic, Google Greely and the topic, and there's a decent chance something will pop up.
Okay, great. So I will be leaving links to your work in the description box of the interview. And it's been a pleasure to talk to you, Dr. Greely. Thank you for taking the time.
It's been great talking to you, and I hope that in a few years when my next book comes out, which thus far is only a title, Playing with Life, about use of CRISPR and other technologies in non-humans, I hope we can do this again.
Yes, of course. Hi guys, thank you for watching this interview until the end. If you like what I'm doing, please consider supporting it on Patreon or PayPal. All of the links are in the description box of the interview. The show is brought to you by people like you, so consider doing it. Otherwise, and if you like the interview, please share it, leave a like and hit the subscription button. This show is brought to you by Enlights Learning and Development Done Differently. Check their website at enlights.com. I would also like to give a huge thank you to my main patrons and PayPal supporters, Karen Litske and Blanchett, Pererga Larsen, Lau Guerrero, Francis Ford, Hans Frederick Sunda, Ricardo Vladimiro, Craig Healy, Adam Kessel, Olaf Alex, Jonathan Wiesel, Jacob Klinkby, Matthew Whittingbird, Arno Wolf, Tim Hollacy, Eric Alenia, John Connors, Pauline Barron, Philip Force Connolly, Jerry Mueller, Herbert Gintis, Rutger Voss, Bo Weingart, Rebecca Newberger Goldstein, Dan Demetrio, Robert Windiger, Rui Inácio, Arthur Coe, Zup, Marco Neves, Colin Holbrook, Susan Pinker, Thomas Trumbull, Bernardo Seixas, Pablo Santurbano, Simon Colombo, Jorge Espinha, Phil Kavanagh, Corey Clark, Mark Blythe, Roberto Nguanzo, Michael Stormir, Eric Neumann, Samuel Andreev, Tiago Nunes, Bernard Yugni, Alexander Dunbauer, Omer Hickson, Fergal Cusson, Evan Bodrenko, Al Herzog, Don Ross, Jonathan Librant, Oslo Bullet, Nathan Nguyen, Stanton T, Samuel Correa, Eric Hines, Mark Smith, J.W. João Eira, Tom Hummel, David Sloan Wilson, Yacilades Araújo, Ethan Solon, Roman Roach, Dmitry Grigoriev, Diego Londonio Correa, Tom Roth, Yannick Punter, Adana Rusmani, Charlotte Bliss, Miran B, Nicole Barbaro, Adam Hunt, Pavel Ostazewski, Max Belby, Nelek Bach, Catherine and Patrick Tobin, Al Ortiz. My producers is Arweb, Jim Frank, Lucas Staffini, Kian Gilligan, Sergio Codreano, Luís Caetano, Tom Vanegdam, Curtis Dixon, João Linhares, Benedict Mueller, Vega Guidi, Sardos France, and Nirvan Balachandran. And my executive producers, Michel Rzeski, Rosie, James Pratt, and Matthew Lavender. Thank you for all.